Thank you very much, Alina, for the invitation, the very kind introduction, and for giving me the chance to present our research here today. And my talk is going to be about ontogeny and anti-metastatic functions of group 1 innate lymphocytes. And I would like to start this talk by introducing our lab, the Innate Lymphoid Cells and Cancer Lab, which is part of the Institute of Experimental Immunology located at the IRHEL campus in the beautiful city of Zurich. And in our lab, we study innate lymphocytes, starting from their origin, the role they play in tissue homeostasis, focusing on different organ niches, and we also study their involvement in pathology, mostly in cancer metastasis. And this is the family of innate lymphocytes, these early immune effectors that carry out specialized functions in tissue homeostasis and in inflammation. And in the lab, we study group 1 ILCs, which include natural killer cells and ILC1s. And the NK cells are the most known and studied members of the family since they were discovered more than 40 years ago. But ILC1s have only been widely characterized in the past few years after they were discovered in the liver as an atypical subset of NK cells. Both subsets provide type 1 immunity against virus-infected cancer cells and as well as some intracellular pathogens. And when we think about how NK cells and ILC1s regulate immune responses, we need to start with immune regulation. NK cells recognize cellular stress ligands and MHC class I and related molecules through a wide range of activating and inhibitory receptors. And the balance between these signals is going to determine whether the target cells will be eliminated or spared. ILC1s don't express most of the Li49 receptors that receive inhibitory signals from MHC class I, but they can still sense non-classical MHC class I molecules via NKG2 receptors, and they can get activated via receptors such as DNMA1 or P2RX7, which recognizes ATP from damaged cells. Both subsets are activated by cytokines, which regulate not only their survival, but also effector functions. And upon activation, NK cells release these cytotoxic granules, such as perforin or granzyme, to kill target cells. And this is shared with certain subsets of ILC1s, which can also kill via trail death receptors. And both subsets have the capacity to release high amounts of cytokines, such as interferon gamma or TNF, which will heighten immune responses. And we know now from many studies that NK cells and ILC1s display considerable overlap and interchangeability, but they are considered different lineages. The main difference between them is that NK cells circulate through the body, whereas ILC1s are resident in some tissues, and this is reflected in the repertoire of cell surface markers. In mice, both subsets are defined by the expression of the receptors NK1.1, NKp46, or CD122, but whereas NK cells feature the integrin CD49B or the Li49 receptors, ILC1s preferentially express markers of immaturity and residency, such as the integrin CD49A, the chemokine receptor CXCR6, or the CD200 receptor. In terms of transcriptional regulation, both subsets depend on TBET for their development, but IOMES is the transcription factor that is essential for the development of NK cells, while HOBIT is the one that controls the differentiation towards ILC1s. And this is only a very simplified scheme where we can add many levels of complexity if we take into account the heterogeneity of NK cells and mainly ILC1s in different tissues. And what do we know about the origin of these cells? The developmental relationship between NK cells and ILC1s has been a matter of debate for a while, but we know that in the bone marrow, both subsets can develop from common lymphoid progenitors and later on from innate lymphocyte progenitors that retain the potential to generate ILC1s and NK cells. And this is orchestrated by a wide array of transcription factors at the different developmental levels. More recently, the groups of Vivier and Tian have shown that ILC1s can also develop locally in the liver from hematopoietic stem cells that give rise to these CD122 positive, CD49A positive precursor cells that have a restricted potential to differentiate towards ILC1s. And despite this bunch of evidence, it is not clear how the origin of these cells affects their fate, and we wondered whether a different developmental origin of NK cells or ILC1s could imprint the functional diversity of these cells in adults. And to answer to this question, Colin Sperano, a very talented PhD student in the team, put together an integrative study using fate mapping, omics techniques, and adoptive cell transfers to investigate the ontogeny of NK cells and ILC1s and how this impacts on their function. Colin first looked at the turnover of these cells in adult mice using ID2 CLIE-RT2 YFP mice, which allow us to label and trace both NK cells and ILC1s upon tamoxifen administration. You can distinguish here very well NK cells, which are positive for the integrin CD49B, from ILC1s, which feature the integrin CD49A. And after tamoxifen, about 30–50% of these NK cells and ILC1s are labeled with YFP. And if you now follow these cells over several months, you can see a fast decay of NK cells in all organs, which is in line with their continuous supply from the bone marrow. But for ILC1s, the picture looks very different. So there is a fast turnover in organs like the uterus, intermediate in the salivary gland, and only a minimal dilution in the liver observed at least until 3 months of age. And this indicates that the population in this organ is very stable. And since this population appears to be so stable, we then looked early in development in the fetal liver, and we found embryonic ILC1s already at E14.5. And in contrast to the adults, these embryonic ILC1s co-express CD49A and CD49B, and they undergo a drastic expansion around birth. We could also visualize these cells by immunofluorescence using NKp46 CRE mice crossed to rosa tomato mice, with most of these ILC1s being associated with the vasculature and forming cell clusters. We also found these embryonic ILC1s in other tissues like the spleen, as you can see here, but at the postnatal period, these cells disappear and they get replaced by CD49B positive NK cells in all organs apart from the liver, where these cells will remain stable. And to better define the features of these cells, Colin performed single-cell RNA sequencing in group 1 ILCs isolated from livers at embryonic day E18.5, postnatal day P10, and adult mice. We could clearly identify 4 clusters of proliferating cells, 2 clusters of ILC1s depicted here in red, which express the CD49A and CD200 receptor, and 3 clusters of NK cells depicted here in blue colors, which feature high levels of the transcription factors ZEP2 and EOMES. And if we follow the kinetics over time, it is clear that ILC1s are established very early in life, whereas NK cells originate and mature much later between the postnatal stage and adulthood. At E18.5, we can observe many group 1 ILCs that are still proliferating, and at postnatal day P10, many of these cells already cluster as mid and late ILC1s, which are both differentiated cells expressing high levels of residency and cytotoxicity-associated genes, with the mid ones retaining some markers of earlier developmental stages. Also at the postnatal stage, we observed the emergence of a new cluster of early 1 ILCs here that expresses early differentiation markers like MB gene, IL-18 receptor, or TCF7, but this cluster was very heterogeneous because it contained cells adjacent to ILC1s, which resemble this helper-like ILC1 subset identified by the Gazzeiger group as cytokine producers, but also cells on the opposite side displaying high levels of NK cell-specific genes, which resemble immature NK cells. We then performed trajectory analysis using RNA velocity to study the relationship between the subsets, and we could identify a first trajectory for the embryonic and neonatal stages, which goes from these proliferating group 1 ILCs towards mid-late ILC1s, and a second trajectory emerging only in the postnatal stage from these early group 1 ILCs towards mid-late ILC1s. And this became the primary path in the adult liver. This short trajectory towards ILC1s is always accompanied by the acquisition of the cytotoxic and residence genes that define the most differentiated subsets. And from this data, we hypothesized that hepatic ILC1s may differentiate as 2 waves, one starting during the embryonic stage and another one arising postnatally. So we first focus on the embryonic wave, and we fate map embryonic ILC1s using the ID2 CRT2-YFP system and analyzed the organs of adult mice. After tamoxifen administration, about 25% of these embryonic ILC1s were labeled with YFP, and in adult mice, we found that around 40% of ILC1s in the liver were still fate mapped, but not in all other organs. And this demonstrates that embryonic ILC1s persist until adulthood only in this organ. We could also trace the wave of embryonic ILC1s using flow cytometry because these cells are positive for the Li49 receptor E, and using this marker, we could see that in adults, these embryonically derived cells represent the most differentiated ILC1 subset, expressing low levels of IL-7 receptor and TCF1, but high amounts of the residency markers in Granzyme. But these embryonically derived ILC1s only represent a 40% of total pool of ILC1s in the adult liver, so the rest may come from somewhere else. We then went back to the second trajectory, arising in the postnatal stage from helper-like ILC1s towards more differentiated cells to investigate if this is a distinct wave of differentiation. Using flow cytometry and the surface markers MB gene and IL-18 receptor, we can distinguish between helper-like ILC1s, which are here positive for both these markers, and the more differentiated ILC1s, which don't express them. We could confirm that these helper-like ILC1s produce more cytokines and lower levels of granzyme B, and they kill less in comparison to the more differentiated ILC1s. And this is consistent to what was shown by the Gasteiger group in adult mice. And to make sure that the helper-like ILC1s arise only in the postnatal stage and not before, we fate mapped embryonic ILC1s again and analyzed the liver at postnatal day P10. And we found that the most differentiated ILC1s here were not diluted because they really come from the embryonic stage, but the helper-like ILC1s as well as the immature NK cells mainly contained unlabeled cells, which confirms that this is indeed a distinct developmental wave. We then asked if these helper-like ILC1s that emerge postnatally constantly seed tissue ILC1s or whether this was restricted to this neonatal window. So we fate mapped group 1 ILCs at the postnatal stage peak 12 and then analyzed tissues of adult mice. And we found that in the liver, the labeling frequency remained stable until adulthood, in contrast to all other organs where ILC1s as well as NK cells were more rapidly diluted. And this shows that these hepatic ILC1s are fully established within the first 2 weeks after birth, while other ILC1 pools are still dynamic at this stage. We then asked if these helper-like ILC1s that emerge postnatally we then wondered about the origin of these neonatal helper-like ILC1s, and we turned to the bone marrow, which contains these precursors for both NK cells and ILC1s. So we performed single-cell RNA sequencing in the bone marrow at postnatal stage P10 and adult mice, and we could identify a small population here of helper-like ILC1s depicted in red, which expresses the classical ILC1 markers, but not the omes. And when we transferred these helper-like ILC1s from the bone marrow into red gamma-C knockout mice, we found that they were indeed able to generate hepatic ILC1s. And this is the recap of this work, where we show that hepatic ILC1s originate in 2 developmental waves. A first wave emerges here during embryogenesis in the liver and gives rise to the more differentiated cytotoxic subset of ILC1s. And after birth, a second wave of helper-like ILC1s and NK cells from the bone marrow will quickly replace ILC1s in most tissues apart from the liver, where they layer with the embryonic subset and persist until adulthood. And we now know that hepatic ILC1s arise in these prenatal stages, but the next question is, how does this work? How is this regulated? And to answer to this question, we focused our attention on the transcription factor HOBIT, which is known to regulate a universal program of tissue residency in lymphocytes. We performed single-cell RNA sequencing of livers from HOBIT knockout mice at these 3 developmental stages and compared them to the wild-type counterparts. We observed a clear decrease of the population of these differentiated cytotoxic ILC1s and higher numbers of the helper-like ones and immature NK cells, meaning that the cells were really skewed towards an earlier phase. And this dependency on HOBIT for ILC1 differentiation was already very evident at the postnatal stage P10, but became much more drastic in adult mice, in agreement to what was shown by the groups of Georg Gasteiger and Marco Colonna. We then wondered how this impaired differentiation caused by the lack of hobbits leads to an almost complete absence of these cells in the adult liver. So we compared the transcriptomes of more differentiated ILC1s at P10 between hobbit wild type and hobbit knockout mice. And many of the downregulated genes, as you can see here in these hobbit knockout ILC1s, were related to mitochondrial respiration and oxidative phosphorylation pathways. So we measured the oxygen consumption rate, which is an indicator of oxidative phosphorylation, and we found that it was much lower in hobbit knockout ILC1s, which indicates that the perinatal differentiation of these cells involves this metabolic adaptation. And another factor that we found less expressed in ILC1s from livers of hobbit knockout mice was the transcription factor TOX, which is involved in T-cell differentiation and exhaustion during viral infection and cancer. And as you can see here, TOX is highly expressed in this population of differentiated cytotoxic ILC1s in comparison to the helper-like ones. And when we generated mice that have a deletion of TOX specifically in group 1 ILCs, we observed a drastic decrease of the cytotoxic ILC1 population, which highlights again the importance of this factor for the generation of this subset. And we know now a bit more about the ontogeny of group 1 ILCs, but what about their function? What's the role of these cells in a pathological setting? NK cells have been studied for many years for their antiviral and anti-tumor properties, but more recently, some studies have also revealed non-redundant functions of the resident ILC1 subset. And focusing only on the liver, a first publication pointed towards a role of this resident population in conferring immunity in skin contact inflammation. And more recent work has shown that ILC1s are capable of providing early protection against viruses, and they can also protect from acute liver injury. And there is a pathological context in which we thought these cells could play a relevant role, and this is metastatic disease. And as you all know, metastasis is the last stage of cancer and the key cause of failure of cancer therapy and mortality, accounting for about 90% of cancer-related deaths. Metastasis is a multi-step process. It starts once the tumor cells invade the surrounding tissue locally. They then intravasate into the circulation until they get arrested and extravasate into target organs, which they colonize. And if they are successful, they will form what is known as a metastatic colony. And during all these steps, the success of these metastatic cells will depend on the interactions with the immune system. And we are especially interested in understanding how innate lymphocytes are regulating the different steps of the disease. And we know for many years that natural killer cells are particularly efficient in controlling metastatic disease. And you can see this here in this metastatic model of breast cancer, in which we inoculate tumor cells in the memory-fed bed and deplete NK cells using an anti-Asialo antibody. 30 days upon tumor inoculation, we observe no change of primary tumor growth, but an important increase in the number of metastases in the lungs. And there is an extensive bunch of preclinical literature which further supports this notion and elucidates part of the molecular mechanisms through which NK cells control metastasis, which involve several activating receptors and also effector molecules. And if we now focus on liver metastasis, the situation is a bit more complex because this niche is rich in circulatory subset of NK cells, but also in resident ILC1s, which may play different roles in the development of the disease. We can induce hepatic metastasis by injecting cancer cells intrasplenically, followed by splenectomy. And we teamed up here with the group of Professor Maries van den Broek in Zurich and used two different tumor models to address this question. The colon carcinoma MC38, which is highly immunogenic, and the less immunogenic Lewis lung carcinoma. We inoculated these two tumor cell lines into NKp46 Cre DTR mice, which allows us to deplete group 1 ILCs at different time points using diphtheria toxin administration. And you can see here that the administration of diphtheria toxin depletes both NK cells and ILC1s very efficiently. And for the metastatic load in the liver, we observed an increase in both tumor models when group 1 ILCs were depleted before or 24 hours after tumor inoculation. But when we depleted the cells later, 7 days after tumor injection, this only resulted in larger metastases from MC38 cells. And this suggests that group 1 ILCs control early metastatic stages in both models, but they are only able to control metastatic outgrowth in the more immunogenic tumor type. And indeed, these MC38 metastatic nodules are more infiltrated with group 1 ILCs in comparison to the Lewis lung carcinoma-derived lesions. And you can see this here in this immunostaining, where NKp46 are marked with NKp46 positive cells, are marked with tomato, and the tumor cells and the endothelium with PCAM1. So group 1 ILCs can control liver metastasis, but how can we check the specific contribution of the resident ILC1 versus the circulatory NK cell subset? For this, we used mice that lack iomes in group 1 ILCs, which don't have NK cells, but ILC1s are not affected. And if we want to get rid of ILC1s but not NK cells, we have to use Hobbit knockout mice. So group 1 ILC, and using this strain, we found that when mice lack either NK cells or ILC1s, the metastatic burden in the liver is increased in both tumor types, meaning that the two subsets are able to restrict liver metastases. So the next question then was, do they really perform the same job, or is there a difference in the way these two subsets control metastatic disease? And there is a difference in the way the two subsets control metastasis, which has to do with timing. And to show this, we used the two mouse models lacking either NK cells or ILC1s, and we additionally depleted the remaining population at different time points using the DTR system or an anti-Asialo antibody. And we could see that only when ILC1s were depleted 48 hours before tumor inoculation, but not later on, metastasis increased in both tumor models, indicating that ILC1s control metastasis at early stages during seeding. However, when we depleted NK cells from Hobbit knockout mice, we observed that, again, NK cells control early stages of both Lewis lung carcinoma and MC38-derived metastasis, but they can only control late stages here in the more immunogenic MC38 model. We also found that ILC1s do not infiltrate the metastatic nodules as NK cells do, and probably because of that, these ILC1s from both tumor models retain the capacity to kill tumor cells, even when we isolate them at advanced metastatic stages. Whereas NK cells from both tumor models became clearly dysfunctional and killed tumor cells much less efficiently than those isolated from naive livers. And since we observed this differential regulation of metastasis in these two tumor types, we then investigated how the metastatic niche influences the phenotype of NK cells and ILC1s using high-dimensional flow cytometry. And to do this, we separate the metastatic nodules from the adjacent tissue, and we compare these two regions to naive livers. The Lewis lung carcinoma model is depicted here, and we identified a CD49A positive ILC1 subset here in red, which does not change much during the course of metastasis, and two subsets of NK cells, one more and one less differentiated, with this less differentiated one expanding during disease progression. We also found a subset which emerged in Lewis lung carcinoma metastatic livers, which is depicted here in this yellow color, and this subset expresses very low levels of the transcription factor iomes and some other markers that are reminiscent of very immature NK cells. We then examined MC38-derived metastatic livers, and we identified another unique subset here of NK cells, which is very enriched in the nodules. And this subset expressed also some immature NK cell markers, but in contrast to the subset found in the less immunogenic tumors, this features high levels of iomes and expresses the resident integrin CD49A. And both this subset and the one found in the Lewis lung carcinoma model were considered to originate from NK cells because we did not find them in NKp46 cryomes flox mice which lack NK cells. And an extra consideration here is that these unique populations were also found in other tumor models with similar degrees of immunogenicity. We then checked for functional features of these subsets, and we found that this intranodular subset of MC38 metastasis produces high amounts of granzyme B in comparison to the low amounts found in the iomes low population of Lewis lung carcinoma-derived nodules. And when we stimulated these cells ex vivo using anti-NK1.1, PMA ionomycin, or a combination of cytokines, we observed higher amounts here of degranulation and interferon gamma production in the MC38-derived subset in comparison to the more immature one from Lewis lung carcinoma metastasis. So it seems that the resident subset from more immunogenic tumors still retains some functional features, which is not the case for the subset that arises from a less immunogenic tumor type. We also performed single-cell RNA sequencing of group 1 ILCs sorted from naive Lewis lung carcinoma and MC38 metastatic livers. And we found a cluster here which was enriched in Lewis lung carcinoma-derived metastasis, which features several markers of immature NK cells, and another cluster here which expresses high levels of CD49A and EOMES, and this corresponds to the population identified by flow cytometry in the MC38 nodules. And when we looked deeper into gene signatures, we found that many of the genes defining this population were related to both TGF-beta and IL-15 signaling pathways. And since this data pointing towards TGF-beta and IL-15 related signaling, we quantified the transcripts of these two cytokines in the metastatic regions and naive livers, and found high amounts of TGF-beta in the adjacent tissue and also in the nodules of both metastatic models, but the levels of IL-15 here were only high in MC38-derived nodules, and the two cytokines were needed in vitro to induce these characteristic features of the resident subset of NK cells. We also crossed NKp46 mice to TGF-beta receptor flox mice to specifically delete TGF-beta signaling in group 1 ILCs. And for the Lewis lung carcinoma model, the metastatic burden did not change, nor did the phenotype of NK cells or ILC1s. But for MC38 metastasis, for the MC38 model, the metastases were clearly reduced in mice lacking TGF-beta receptor on group 1 ILCs. And this correlated with the disappearance of the CD49A positive EOMES positive NK cell subset from the metastatic nodules. And this is how we think metastases modulate group 1 ILCs in the liver. The resident ILC1 population appears to act as gatekeepers and eliminate tumor cells when they enter the liver. And the role of NK cells is much more influenced by the degree of immunogenicity of the tumor type, with these less immunogenic tumors favoring NK cells that lack the expression of the master transcription factor EOMES. They cannot infiltrate the nodules properly, and they are immature and very dysfunctional, whereas more immunogenic tumors feature a subset that infiltrates the nodules, acquires residency features, and although it cannot properly fight the tumor cells, it still retains some functional properties. This subset, which is induced by IL-15 and TGF-beta, is very reminiscent of an ILC1-like subset, which was described by the group of Mark Smith in fibrosarcomas, which was not able to control tumor growth, but also of a subset of ILC1s that arises in breast cancer tumors, which is expanded by IL-15, and in that case, contributes to anti-tumor immunity. But this bunch of data arises from experimental mouse models, and the question that comes up is whether we can translate these findings into human metastatic disease. In humans, the liver is one of the main sites of metastatic spread, representing about 25% of total cases, and about 50% of patients with colorectal cancer, for example, will develop metastasis, being the liver the most common site. And when this happens, hepatic resection is still the only realistic cure, but even when the liver is resected, only 30–40% of the patients survive in 5 years, and current immunotherapies don't seem to be very beneficial. So there is a real need for additional targeted therapies and prognostic biomarkers to stratify the patient's outcome. And we thought that we could contribute to this by mapping immune cell signatures that could be of therapeutic or prognostic value for liver metastasis. And to do this, a brilliant team from the lab, headed by the PhD student Maud Mayu, generated a tissue biobank of liver metastases from colorectal cancer patients in collaboration with the Department of Pathology at the University Hospital in Zurich. These patients have similar characteristics and present with microsatellite stable CRC. The liver was resected within 1 year after diagnosis, and they underwent treatment with neoadjuvant chemotherapy or targeted therapy. And we have collected these 3 representative regions, which were also used in our mouse models, including the nodules, the adjacent tissue or invasive margin, and also distal tissue to the nodules. And we have collected them in different sample formats. And using cryopreserved cell suspensions, we performed multiparametric flow cytometry covering these 3 metastatic regions and using large panels of lineage and activation markers for different immune cells. And we could identify different lymphocyte subsets, as you can see here in this UMAP, including 2 populations of NK cells consisting of the more immature CD56 bright and the cytotoxic CD56 dim. And when we looked at the distribution of the cells in these 3 metastatic regions, it was very obvious that both subsets are clearly excluded from the metastatic nodules, And this is also the case for any other subset with cytotoxic features, including effector T cells, myeloid, or gamma delta T cells. On the other hand, the metastatic nodules are highly enriched in memory populations of CD8 and CD4 T cells. And we then zoomed a bit further into the NK cell population, and we observed the emergence of a subset here, which is depicted in blue, which expresses EOMES, residency markers, including CD69, CD103, and also CD49A, and intermediate or low levels of granzyme B. And this is very reminiscent of this resident subset, which we found in metastatic livers in mouse models. And when looking at the different areas of these metastatic livers, we could see that while the two subsets of immature and cytotoxic NK cells were very much absent from the metastatic nodules, this subset with these residency features is clearly enriched. We then tried to correlate the abundance of this resident subset with response to neoadjuvant chemotherapy, but found no clear correlation with the tumor regression grading, which is the measure of histopathological response to treatment. And we are now currently exploring further features of these subsets and also the correlation with other clinical parameters, like, for example, survival, time to relapse, and the mutational status. And there are many research questions we still want to tackle. We are mapping other immune signatures of colorectal liver metastasis with prognostic and therapeutic value that we can further explore in reverse translational studies. We are also very interested in deciphering the niche-specific factors that regulate ILC functions in both homeostasis and metastatic disease. And finally, we aim at understanding the mechanisms that lead to NK cell paucity and dysfunction in the metastatic sites. And I would like to finish here by acknowledging all these wonderful members of the innate lymphoid cells and cancer team who are working very hard on these projects and also some other ongoing projects in the lab, our collaborators at the Department of Pathology at the University Hospital in Zurich, the members of the Institute of Experimental Immunology, and all collaborators in Switzerland and abroad for the great help. And very importantly, I would like to thank the funding agencies and all of you for your attention.